All right, sounds good on our end. So before we get started, I just want to say our apologies we couldn't be there in person today. It's a little bit of a hectic schedule for everybody on the team on our end, so sorry for that, but hopefully it doesn't take away too much from the presentation. And with that being said, we'll jump right into our presentation. All right, so getting started, just a general brief overview. As I'm sure all of you are aware, gliomas are a very, very common malignant brain cancer that's both aggressive and often reoccurs. And so for this reason, we decided that this is going to be something that we're going to look into. So going forward, one of the obstacles in selecting a treatment target is tumor heterogeneity, as neighboring tumor cells may display distinct morphological and phenotypic profiles. Thus, when selecting a target, the team had to choose a cellular aspect that is crucial to cell survival and therefore remains fairly constant across cells. After reviewing recent literature, we believe targeting mutant isocitrate dehydrogenase is both a viable and realistic treatment option. In healthy cells, isocitrate dehydrogenase is an enzyme that functions in the citric acid cycle and is essential to create molecules that are used for cellular energy. This is done through oxidative decarboxylation that yields NADPH. There's two isoforms that naturally occur, IDH1 and IDH2, but for the focus of our presentation, we'll be focusing on IDH1 that plays a predominant role in lipid metabolism and glucose sensing. Now, interestingly enough, in 2008, a study sequenced more than 20,000 genes from 22 GBMs and identified a shared point mutation in IDH1 at arginine 132. On the right, we can see a crystal structure showing mutant and wild-type isocitrate dehydrogenase 1 active sites. Note that the histine in place of arginine 132 in the mutant. In all, a shared point mutation in IDH1 at this location was found in around 12% of the samples. Subsequent studies determined this mutation was present in about 80% of grade 2 and 3 gliomas and secondary GBMs. So this ultimately accumulates into a gain of function. IDH1 now produces 2-hydroxylglutarate. So what is 2-hydroxylglutarate? Well, it's a mutant IDH produces this oncometabolite that has numerous epigenetic effects on the common glial precursor cells and is likely a prerequisite for the 1p19q codeletion that subsequently leads to tumor development.
So how does 2-HG go about causing cancer? So there's three main hypotheses as to how this happens. The first being that 2-HG acts as a competitive inhibitor of DNA and histone modifying enzymes. So as you're aware, these kind of epigenetic modifications are really critical in determining everything from cell cycle progression to apoptosis, to rest, all that kind of stuff. So any aberrant epigenetic changes are going to increase the risk of cancer and differentiation blocks specifically. So the second hypothesis posits that 2-HG acts as a competitive inhibitor of prolyl hydroxylase. So normally, hypoxia-inducible factor is degraded via hydroxylation of its proline groups. But obviously, if prolyl hydroxylase is being inhibited, this can't occur. So excess HIF1A is going to accumulate, and this induces increased vascular endothelial growth factor, and this in turn leads to angiogenesis, which is a requisite for tumor development. So the third hypothesis is a little bit simpler, and it puts forward that DNA damage results because of the lack of production of NADPH, as seen in that original reaction scheme of the isocitrate dehydrogenase mechanism. So without that NADPH to suck up free radicals, they're going to be free to damage DNA, which can eventually cause mutation resulting in cancer. So this IDH mutation seems like a fair smoking gun. So people have definitely tried to drug it in the traditional sense of small molecule inhibitors of the active site of the enzyme. These two structures are a couple of recent examples from a Nature paper that actually inhibit it quite well. The thing is, IDH is such a widespread metabolic enzyme in the body, so any off-target effects are likely to seriously jeopardize its efficacy. use of people. So this is where our solution comes into play. We suggest inhibiting mutant IDH1 at the translational level. So what that means is introducing small antisense oligonucleotides in a cell that have perfect complementarity only to the mRNA of the mutant IDH1, but are not sufficiently complementary to anneal to the wild type with the arginine. So if you get the perfect annealing that we're hoping for here with the small molecules of DNA, it's going to recruit RNAs, which eventually degrades the mRNA, resulting in no production of the mutant IDH enzyme.
This method, as mentioned before, we're planning to use RNAs to degrade the mutant IDH1. RNase H will specifically degrade the RNA-DNA hybrids formed by the binding of the antisense and mutant RNA of MIDH1. This form of RNase is naturally found in cells for genome maintenance and R-loop processing, and is active in the cytoplasm and the nucleus. This method relies on the use of incorporated DNA to bind to the mutant RNA. This DNA, as shown above, will be complementary to the MIDH, but will be able to distinguish between native and mutant through the codon highlighted in red. In order to ensure the DNA isn't degraded upon entering the body, it will be incorporated by locked nucleic acids, as shown in the picture above, to improve stability. And finally, it will be conjugated by triatenuary and acetylgalactosamine. In terms of the rationale for this experiment, it's first important to understand the relevance of the mutation. In 2008, a study of 20,000 genes and 22 glioblastomas were sequenced and found that 12% had the IDH mutation. But additionally, in a separate study, 80% of grade 2 and 3 gliomas and secondary GBMs found that this mutation also persisted. Therefore, it's a relevant field to put research into. Moving on, this research is also therapeutically relevant by its design. The RNA-DNA hybrids are designed to have perfect complementarity to ensure that there is no danger to native IDH, which is critical for function. In addition, this particular pathway has a lot of potential because this mutation is key driver in a variety of cancers and shares this conserved point mutation. Therefore, it seems like this has a lot of signs of this specific point mutation, and it would be a waste to ignore them. And thanks.